Okay, so now that we know DNA is the heredity material, let's get into the gory details of what it is built out of. So I'm starting from the point that you know that the subunits, the basic subunits of DNA are nucleotides. So DNA is a polymer. We talked about this before, that there's hundreds of millions of base pairs that are in a chromosome. You know, a genome, the human genome has 2 gigabases of base pairs, and so what that means is that the nucleotide subunits, there's a huge number of nucleotide subunits, and so it's important that you understand each of the components of a nucleotide. So there's 3 main components. There's a nitrogenous base, there is a sugar, and then there is a phosphate group. And so make sure you're able to recognize each of these components, so the sugar and then the base. So the nucleobase is important because that provides the variability of a nucleotide. So there's 4 different kinds of nucleotides, and there's 2 different types of nitrogenous bases. So we'll get into those details in a second. But there's pyrimids, there's purines and pyrimidines, and then there's also A, C, Gs, and Ts. And so this is where the variability of DNA comes from. So this is how it's able to encode information, is that it's each nucleotide provides 4 bits of information. The sugar is also important. So this is the 2-deoxyribose sugar. And so like many sugars, but not all sugars, it's a pentose sugar. And the basic part of the sugar is a ribose sugar, but it has a unique modification at a specific carbon. So instead of it having an oxygen, a hydroxyl group bound here, it does not have that oxygen, and so that's where the 2-deoxyribose comes from, because it's in the 2' position of the carbon. And then finally the phosphate group right here. So this phosphate group is important because it provides the negative charge on DNA, and it is in response, and consequently is what gives the acid for part of this DNA. So DNA is an acid, and the reason it's an acid is because of the addition of the phosphate group. So first, let's talk about the sugar, the 2-deoxyribose sugar, and this serves as part of the backbone of the DNA. And so if we make, double-stranded DNA, the backbone, there's the backbone, and then there's also kind of connections in between the backbone. So the sugar and the phosphate both make up the backbone. So these are carbon molecules. So there's 5 important carbon molecules. So the top of the sugar is an oxygen, and then there's 1, 2, 3, 4, 5 different carbon molecules that are part of the 2-deoxyribose. So it's very important to remember the order of these carbons. So start from the oxygen and then go clockwise. The 1, 2, 3, 4. So the 1 to 4' are part of the pentagon, and then the 5th carbon is outside of the pentagon. So it's 4 of these carbons have very important roles. So the 1' carbon, so it's important to remember this because this is where the nucleobase binds, so the connection point between the A, the C, the G, the T part of the nucleotide occurs at the 1'. The 2' is important to remember because this is what distinguishes DNA from RNA, so whether or not this group right here is a hydrogen atom or it's a hydroxyl, like an OH. So in RNA it's OH, in DNA it's just H. And then the 3' and the 5' carbons are very important because this is where the nucleotides polymerize together. So, we talked about nucleotides being covalently bond together. So the upstream nucleotide would bind here, and then the downstream nucleotide would bind here. And so this also provides an ordering of the chain, which we'll go over in a little bit, in the 5' to 3' direction. So if you've ever heard someone talk about DNA and talk about the 5' to 3' direction, this comes from the naming system of the carbon atoms within the sugar. Nitrogenous bases. So there's two kinds of nitrogenous bases. So this is what distinguishes nucleotides from each other. And they can either have purines, so these are 9-member rings, or pyrimidines, and these are 6-member rings. So the adenine and the guanine are the A and the G. The cytosine and thymine are the C and the T. And the uracil is U. And so uracil is part of RNA, T is part of DNA, and then C is part of both RNA and DNA. So each of these have a different nitrogenous bases. So these, unlike sugars, are very rigid molecules that are planar, and that's due to double bonds between the carbon atoms. And so you don't have to memorize the numbering of these. But you should be able to distinguish the pyrimidine ring and the purine ring. So just remember that the pyrimidine ring, the word is longer, but it's a much smaller molecule, so it's kind of opposite. The purine is the smaller word, but it's a much larger molecule. So I'm showing you the numbering system here, but I don't expect you to memorize that for the class. But I do expect you to memorize cytosine, uracil, and thymine are all pyrimidines, and then guanine and adenine are all purines. So it's important that you memorize that. It's also important to memorize the location of the nitrogen atoms that are used to create the covalent bond with the sugar. So where do these nitrogenous bases bond to the sugar? You should also memorize that information. So as an example, we're going to take this nitrogenous base. This is the guanine. And so how does this form, how does this connect with the deoxyribose sugar? And so we talked about before, the 1' end right here is able to bind to the nitrogen atom on the guanine. And so as a result of this bond, you actually lose a water molecule. So the OH group on the carbon and the H group on the guanine bind together to form H2O. And so that also creates a water molecule. And so basically, carbon wants to have 4 bonds, and so it loses 1 bond here, and it gains this bond here, where nitrogen wants to have 3, so it loses this bond here and gains that. So that's the way it's able to keep the number of bonds the same, is by actually creating this water molecule. And so it's also important to note this is where the word nucleoside comes from. So a nucleoside is a molecule that's a combination of the sugar and the nitrogenous base. So it's not a nucleotide because it also lacks one of the 3 key components. So just to reiterate, a nucleoside contains a nitrogenous base and a pentose sugar. And a nucleotide also contains a phosphate group. And so the nucleotide is the subunit of DNA. And so finally, the phosphate is attached to the 5' carbon. So this is where it's important to remember this numbering scheme. So this phosphate group attaches at this carbon, and because the phosphorus has 5 bonds, so this is a double bond, a single bond, a single bond, a single bond, instead of 4, it's acidic in nature. It has a negative charge because of it. So the phosphate is attached to the 5' carbon. The nitrogenous base is attached to the 1' carbon. These are double bonds, so you should always remember that. The nitrogenous bases also have double bonds, and that's what creates them. That's why they're very rigid. So they would be like a plane, and they would be very fixed. Whereas the sugar, it's kind of, it has a lot more ways to rotate around, and so it's very flexible in nature. And so what this means is that the backbone is very flexible, so you can move it, you can twist it, you can rotate it. There's a lot of degrees of freedom, but the nucleobase, so the inside, which makes the connection in the double-stranded DNA, that's very rigid. And so what it means is you're trying to line up 2 very rigid items together. So each chemical molecule has a unique name. So if we look at the nitrogenous base, or the nucleus base, adenine, cytosine, guanine, thymine, if we make it into a nucleoside, so we might want to distinguish between ribonucleosides and deoxyribonucleosides for DNA versus RNA. And so you'll notice that we've changed the adenine to adenosine. So we've added a sine here, and we also added the deoxy. So because this adenine can either be bound to a ribose sugar or a deoxyribose sugar, this is just telling you this is part of DNA. So both of these naming schemes are for DNA type molecules. The cytosine becomes cytidine, guanine becomes guanosine, guanine becomes guanosine, and then the thymine becomes thymidine. And then of course the deoxy is appended to above. And then if we add the phosphate group, so we've made the nucleoside acid, because remember the phosphate is why it's acidic, and so we then also add an acid to the name. So deoxyadenylic acid, deoxycytidyllic acid, etc., etc. So you know, when you're going through these, it's important to you know, you might be reading something and it might be a little bit hard to memorize all this stuff, but you know, it's important to remember if you're reading something and they refer to cytidine or cytosine, that you have to think to yourself, okay, is this talking about the nucleobase, or is this talking about the nucleoside, or is this talking about the nucleotide? And then of course you can always Google the word to see what exactly it means. There are also different forms of nucleosides that either have the monophosphate, diphosphate, or a triphosphate added to it. So a nucleotide is a nucleoside monophosphate, but you can also add an additional phosphate group. So if you had two phosphate groups, it would be diphosphates, and then a nucleoside triphosphate would have three phosphate groups. So the nucleoside triphosphates, these are important to remember because these are the actual precursor molecules that are used by DNA replication. And so, you know, when you're incorporating a new molecule, a new nucleotide, or a new base during DNA replication, the final form, you know, will look something like this. Where you have each nucleotide has only one phosphate, but the precursor molecule that gets added will actually have three. And so this would be incorporated right here, and these two phosphate groups would be cleaved, which provides the energy to add to the bond. So we'll go into this in more detail in the next chapter. But just it's important to remember that why we distinguish between these is because this is the precursor molecule. The nucleoside triphosphate is the precursor molecule that eventually gets incorporated into DNA. And so, you know, the extra phosphate groups are just appended to the phosphate molecules. ATP, adenosine triphosphate, this is also an important molecule. So again, if you look at here, this is the 2', so this is not a DNA. So you can always look at these and distinguish, am I looking at a ribose or a deoxyribose sugar? So on the left, we're looking at DTDP, so this is, you know, the nucleobase is different here, it's a T versus an A, but you might, but you also should note that this sugar is a ribose sugar. Adenosine triphosphate is also very important to the cell, so this is also something you obviously covered in your intro bio because it provides the energy source for the cell. All right, so that covers part 2.